and welcome to another Pype pod. This lesson here we're going to be looking at the vessels and how the vessels work in order to assist our body by delivering nutrients and removing waste, and the role that the heart cardiac output plays in the overall perfusion status of the body. That's the end goal of this particular presentation. So looking at the vessels themselves, as you mentioned in the last one that the whole network was a system that starts at the heart and ends at the heart. So that's what the vessels do. They exit the heart with the arteries, okay, and then they travel all around the body, and then they come back to the heart itself. So the vessel tissues starts with arteries, moves into arterioles, then we go into capillaries, then venules, and then back to veins. Now each one of these are uniquely shaped in order to support their function and their structure itself. Starting with looking at the vessels themselves. Now the vessels, the larger vessels, arteries, veins, arterioles and venules, are broken up into three different layers, each one of them starting with the tunica. Okay, so working from the in out, we'll start with the tunica imita, which essentially is a endothelial layer that is just protective layer, slightly connective tissue, and it protects itself from the next layer, which is the actual muscle layer. Now what's unique about the tunica imita is if it was by itself, it's actually extremely permeable. Now this is the cellular structures that are responsible for releasing a lot of those cellular mediators that we spoke about before that help with clotting and inflammatory processes. These are all found in the endothelium layer of the tunica imita. Now the next one down is the tunica medium, which is the muscular layer. Now if you look over at the artery side, you'll see that the muscular layer of the artery is much larger than the vein. Now there's a reason for that is obviously the artery has to be able to contract a little bit harder than the vein in order to maintain the pressure needed to travel through the body, in order to keep up perfusion. It has to have a greater effect on vasoconstriction when we're dealing with getting to the capillaries and allowing for perfusion. Therefore it has a much larger muscular structure. Now the external layer, the tunica externa, is fibrous mesh that holds it all together, stops the media from falling apart, and keeps it from overexpanding itself as well. Now the internal aspect of each vessel is called the lumen. Now a lumen is just a highway, okay, just a traveling highway, and that's it. Once we get down into the capillaries themselves are just the tunica imita. Okay, so they're a endothelium layer, and they have what's known as a basement membrane, and that is just a really thin permeable membrane that allows those cells to hold their shape. And that's it. So that's basically the vascular structure as they're broken down. When you look at each one differently though, you'll see that they have unique structures that allow them to do their job. So starting right at the top in the arteries, we have two different types of arteries in the body. The first one is elastic artery, and then we have the muscular artery as well, hence why elastic and smooth muscle is quite high in both. Now the elastic arteries are mostly found around the heart itself. So this has to allow for the ability to take a huge amount of pressure straight from the heart. So it has the ability to stretch, to accommodate for that pressure, and then move into the muscular arteries that can work with vasoconstriction in order to maintain a relatively good pressure to function. Now the difference between the vessels themselves is obviously the arteries are more muscular, the arterioles lose their elasticity, and they no longer require the fibrous tissue to hold it in place as well, because it's just the push in the capillaries. Capillaries are nothing but endotheliums. And then we move into the venules and the veins, venules, endotheliums, and fibrous tissue. Okay, so that actually allows for venules to have the ability to transport fluid as well. Okay, so capillaries and the venules are the only ones that can transport fluid. Capillaries are the only ones that can transport nutrients. Now the veins are huge. Okay, 5 millimeters in diameter. They're a large, flexible vessel. And the reason for that is they do not have the muscular structure and they do not have a lot of pressure in them as well. So they need to be large in order to reduce resistance within them, so allow for free flow through them. Now obviously the vein is set up with some slightly more unique structures that we'll look at soon, but it's the reason why they're so much larger than the arteries is the arteries are associated with maintaining pressure to optimize perfusion. Veins are associated with being the highway back to the heart in order to start the cycles again. Very low pressure. Okay, when it gets to the heart, we're looking at 15 millimeters of mercury found in a lot of veins. That's all there is. So we need that backfill into the heart, and we need a large amount optimized preload in order to hopefully get a better stroke volume, to hopefully get a better cardiac output. That's why veins are the way they are. Other things that are unique about veins, despite the fact that they're large, very flexible, very moldable vessel, is they have valves. Okay, so the veins themselves have valves. The venules do not. The venules are just pathways post capillaries to move things in. Okay, but the idea of the valves, as this part of blood moves up into the next section of the vein, it stays there. It doesn't allow for backflow. Okay, so that's fantastic. So the whole idea is each time the heart beats, you'll push a little bit more fluid, a little bit more, build it up, and that will naturally allow for the return. But obviously that's not going to be enough, and we've got a lot of factors like gravity and height and size fighting against it. So we need to work on something else. That something else is something known as the respiratory pump. So every time we breathe in, we actually increase the pressure associated in our thoracic cavity. From there, that actually milks the veins in the cavity to bring blood back to the surface. Now this happens all over the body as well in what's known as the muscular pump. Now mostly in the limbs and extremity and along the back. As you contract those muscles, it will literally push against the soft vessels and milk the fluid back towards the heart. Now veins are also capable of vasoconstriction. Okay, so they can constrict in the presence of reduction in pressure or reduction in movement. They can constrict themselves in order to reduce the size of their lumen, in order to increase the pressure and flow back to the heart. So that is what's quite unique about the veins themselves. So they're pretty cool all around. From there we start looking at capillary beds. Now remember I mentioned that capillary beds are the only place in which nutrients can be transported. That's it. So any form of molecule, protein, obviously gas, just at the capillary beds themselves. So the capillary beds come from arterioles and leak into venules. Okay, now capillary beds are made up of two different types of capillaries. So we have the actual vascular shunt, and then you have the capillary bed itself, or true capillaries, which is where the exchange happens. So the vascular shunt is if the area of the body doesn't need any capillary action, then blood can go straight from the arteriole to the venue and then continue without going through the capillary bed. Now this sort of thing happens when you're cold, when the sympathetic nervous system kicks in, vasoconstriction kicks in, certain chemical mediators that tell you that that part of the body isn't being utilized as much, certain muscles, certain organs when you're relaxing. So you don't always have a huge amount of blood flowing all over your body. Okay, if you did, you'd be pretty lightheaded. We have to optimize certain areas of the body in order to do it. Now when waste products build up in that area, then a chemical signal will be released in order to open the sphincters themselves. Okay, so the pre-capillary sphincters found on the arterial side of the house will open, allowing flow into the true capillary beds. Now a capillary bed can be made up of 10 to 100 capillaries depending on the vascularity of the area. Now obviously it is designed to have optimal diffusion out into a greater mass to reach all the cellular structures within it. It also slows down the blood flow completely Obviously, it's much, much slower to get from point A to point B if you're going through the capillary beds, and that optimizes nutrients and fluid exchange within those capillaries. So this is a pretty cool structure that not many people pay a lot of attention to, but it's the reason why we have vascular shutdown. Okay, so everything comes central, all these things we keep saying in signs of shock and when you're cold, all those sorts of things happens without anyone really understanding why that's occurring. And now you have the answer. This is why. Now, looking at the exchange of the capillaries. Now, it's very, very cool. Now, there's a whole lesson on fluid exchange and the concept of diffusion versus osmosis and things like that. So we're just going to give you pseudo cliff notes on this aspect in this one, just to go through what's important. Okay, so as I mentioned, capillaries are responsible for the diffusion of molecules. Okay, whether it's oxygen molecules or nutrient molecules, whatever it is, it's their job. Now, molecules work on a process of diffusion, where they are either drawn by the concentration gradient or the ionization gradient. Okay, so two different structures that can force molecules to move, whether it's high concentration or low concentration, or against its opposing ionization. So positives moving towards a negative environment. That's essentially how molecules will move from the capillaries into the surrounding tissues. Now, different ways it can get through the actual capillary into the tissue is it can obviously just do direct diffusion. That is when you are going from a high concentration to a low concentration. This is what happens when we're dealing with oxygen exchange. Lots of oxygen in the capillaries around the arterial end, not a lot of oxygen in the tissues, so oxygen moves, and vice versa with carbon dioxide. Easy done. When we look at things like the intracellular cleft, that is a pore for fluid-like substances to move through. So a lot of your electrolytes, things like that, can move through these pores, and that's happens. Now, we can have special endothelial fenestrated pores within the structure that allow for special ionized channels. So ionized channels that allow certain structures to get through. That happens at diffusion pores themselves. Now, if something is too large for any of those other means, like a protein, let's say a tissue needs a protein from the blood, then that protein can be boxed up by the endothelial cells into what's known as a vesicle. The vesicle can pick it up, package it, deliver it through the endoplasmic reticulum to the Golgi apparatus, and shoot it out into the cells itself. So that's how capillaries all around work on exchanging the fluid aspect of movement within the capillary cells. What happens is we saw the capillary bed, and you saw the arterial end and the venous end. Now, there's two very different aspects between the arterial end and the venous end. Obviously, the arterial end is going to come into the vessel with more pressure than it would when it leaves the venule end. Obviously, especially if it's gone through the capillary bed itself, it will significantly reduce in overall pressure. Now, when I talk about pressure, there's actually two pressures that are at play when we're dealing with fluid shift. We have something known as hydrostatic pressures, and then we have osmotic pressures. Hydrostatic pressure is just that, the pressure of fluid physically in the blood. So it is going to come through a lot harder and a lot more pressure from the arterial end than it is going to leave the other end. Now, what happens there is fluid is pushed, literally pushed out because of a concentration gradient, out into the interstitial fluid. So from the capillaries, your plasma will be pushed out into the interstitial fluid. Okay, so your hydrostatic pressure at the capillary end is about 35 millimeters of mercury. Okay, the pressure in the exterior is not that. Okay, it's around 10. Okay, or a little bit less, depending on the interstitial fluid that you're dealing with. So it's obviously going to move from a high concentration to a low concentration. Now, the overall osmotic pressure of your blood sits around about 25-26 millimeters of mercury. Now, the reason for that is because of large proteins that are found in the plasma. your albumin, your globulins, your fibrogen. It's found in your blood and it can't get out. It doesn't have the ability to bust through the capillary walls with ease, so it sits in there. The difference is we have a very low net pressure of osmotic pressure in the interstitial fluid because there's not a lot of protein. Okay? Not a lot of protein at all. So at the venous end, we have opposing forces that is actually going to draw fluid in to itself. Okay? Hydrostatic pressure pushes it out. Osmotic pressure draws fluid into itself. It attracts fluid towards itself. Now that is what osmosis is. That is actually the difference between diffusion and osmosis. Diffusion will come in and it will move fluid from a high concentration to a low concentration. That's what diffusion does. The osmosis itself is working on that drawing power. So you have a large osmotic pressure on the venule side, it will gently draw fluid into itself from the interstitial. Now what will happen is you'll get an overall balance, realistically. Now you will lose a little bit of fluid from the capillary, but it's push out and then reconnect and then continue moving. And this happens all the time across every capillary, everywhere. Now obviously when things go wrong and we have a huge amount of hydrostatic pressure, then we can end up with things like edema. If we have a tiny osmotic pressure, then you can end up with things like edema. If you have a huge osmotic pressure, then you can end up with things like dehydration and that sort of thing. Okay, so this is how your hydration status really works and how the capillaries play a very strong role in it. Now, any excess fluid that is left behind in the interstitial space can, if it's too much for the interstitial space, can be picked up by the lymphatic system and then dumped back into central circulation, like we talked about in the last lesson. Okay, but this is fluid movement within the vessels itself. Diffusion and osmosis, two opposing pressures working together to optimize homeostasis. That's what it is. Now, we've mentioned a lot about pressures themselves. So in order for perfusion to occur, there needs to be an aspect of blood pressure. There has to be pressure within the blood. So volume is one thing, presence is another, but pressure is very important in order for ions to shift. If you do not have pressure behind it, you will not have a hydrostatic pressure high enough, and you will not force that ionized shift of a lot of these molecules. So pressure is very, very important. So everything we're learning so far is how to optimize blood pressure when it's needed. So looking at blood pressure itself, it measures the pressure associated with the arteries. So when we look at systolic, that is the highest pressure you can get right at the peak of the ventricular contraction. Now, the diastolic pressure is the pressure that is found within the vessels. Once the vessels have relaxed and the remaining pressure found in the vessels under no external force is what's known as the diastole. Okay, now the pressure found in these arteries is not the same all over the body. Okay, the further you get away from the heart, the less blood pressure you have. And realistically, by the time you get back to the right atrium, the millimeter of mercury blood pressure is around 0. Now, here's a graph that sort of demonstrates that, is as you move from the aorta to the arteries, to the arterioles, to the capillaries, your blood pressure drops. Now, when we talk about clinical blood pressure and everything that is measured and everything that is recorded is looking at the pressure associated with a brachial artery. So if you choose a different vessel, you can expect different results. Okay, so keep that in the back of your mind whenever you think, I'm just going to put this on someone's leg to measure their blood pressure. You will get one, but it will be different. Okay, so just accommodate for that side of the house. So that is how blood pressure works. And clinically, we measure it at the brachial artery. Just remember, it does reduce, and it is all about the power of the ventricle contraction associated with the brachial artery is the blood pressure that we measure. So measuring blood pressure, if we have a blood pressure of 120/70 and we measure it on the brachial artery, what we do with the sphygmomanometer is we pump it up over 120 in order to cut off that arterial supply. So now there's no arterial supply to the arm because the pressure is greater than 120. as we reduce it down and it hits the 120, we have that much pressure, millimeters of mercury, in order to bust through. Okay, now initially, if you leave the cuff on a long time, you get a lot of backfill, a lot of afterload, then it will increase. You will have actually a false high blood pressure because of the pressure that you build up yourself. Okay, but either way, you'll keep hearing the vibration against the vessels until you get down to the resting pressure, which is 70. Once you get down to the resting pressure, which is 70 and below, then there'll be no sounds at all, because the blood will just be stagnant at that stage. You'll feel the pulse of the 120, but you will not hear any vibration against the blood pressure itself. Okay, so that's how we measure the blood pressure. Obviously, there's a huge amount of variations, massively huge amount of variations. Now, a normal range, I'm not going to tell you, you can read it. The term hypertension means obviously low blood pressure. Now, this can happen a few ways. Obviously, it's highly associated with someone being sick, dehydrated, injured, loss of blood. You can also get orthostatic blood pressure. Basically, when you stand up, you lose that blood pressure. And then you'll get like a chronically low blood pressure in a lot of athletes. They don't need a lot of millimeters of mercury in order to function. Okay, now, hypertension occurs, and again, for a whole bunch of different reasons. Okay, exercise, diet, sex, stress, all that sort of thing. You will have a higher blood pressure based on your body. Now, when it happens chronically, you have a chronically high blood pressure, then you're putting your body under a significant amount of strain. So let's say that you're overweight. If you're overweight, you're carrying a lot of adipose tissue, which means you need more vessels, which means you have that longer resistance, peripheral resistance around the body, because the vessels have to travel further. Now, with that, you get an increase in blood pressure because of the peripheral resistance. Okay. Other things that affect peripheral resistance is obviously your blood volume and your blood's viscosity. So how much blood is actually in your plasma. That's an effect that can happen. The more blood you have in your plasma, you can expect a higher blood pressure, because solid matter is going to have a higher pressure than liquid. So that's how variations of blood pressure work. Blood pressure control. Short term versus long term. We have an inner circle and then an outer circle that just grows a little bit bigger. Okay, so looking at the inner circle, let's say your blood pressure drops. Okay, your blood pressure drops, the baroreceptors pick it up and go, we actually need a little bit more perfusion. So the sympathetic system kicks in and we get vasoconstriction. That's what happens initially, but then we also enhance that vasoconstriction with the release of adrenaline and noradrenaline. Now that increases the cardiac output and increases peripheral vasoconstriction, so therefore resistance, increasing the blood pressure. So overall, we have fixed the one problem by increasing the pressure associated felt on those vessels. That's the short-term fix. The problem with short-term fix is if you all of a sudden have chronic hypertension or chronic hypertension, then your body and your baroreceptors are really willing to accept that this is now the new you. This is what's known as an acceptable new level, and it will raise or lower the parameters associated with the baroreceptors. And that's what they do. The baroreceptors will get comfortable with a certain amount, and it's evident with people who gorge themselves as well. Baroreceptors are found in the stomach. You start gorging yourself, you're really full, but then after a while, the baroreceptors get more and more and more acceptable and allow more and more room, and then it goes in. Yes, your stomach grows a little bit, but not as much as people think. It just basically is a changing of these baroreceptors that allows more food to come in. Now, the long-term fix. This is the big one. It's all got to do with the kidney. The kidney plays a direct and an indirect role in the regulation of blood pressure. So if you have a low blood pressure, you're going to have a reduction in the filtration rate through the glomerulus because the pressure's not there. So therefore, you lose less fluid, and that itself stops the blood pressure going lower. If you have a high blood pressure, you have increased filtration, and it sometimes solves the problem as well. On top of that, the kidney is responsible for releasing hormones. Yep, the hormones for the short term and the hormones for the long term. So the adrenal gland itself found on top of the kidney releases adrenaline and noradrenaline, whereas the kidney itself releases renin and it releases erythropoietin, which we spoke about during the blood lesson. And that obviously increases the amount of red blood cell formation there is, which increases blood volume. What renin does, renin is a really stimulating hormone that's associated with the angiotensin action. Okay, so angiotensin 2 is stimulated by renin release. Now, when you're dealing with children, renin is actually the main contributor for your peripheral vasoconstriction, not adrenaline. Okay, so it's actually quite funny. And as we get older, we rely more on adrenaline and noradrenaline, but renin still plays a longer term goal in it. It releases angiotensin 2. Angiotensin 2 increases heart rate and obviously causes vasoconstriction. That's what it does. But what else it does is it stimulates the release of aldosterone, which stimulates the release of your antidiuretic hormone. Now, aldosterone, one of the things aldosterone done is it actually holds on to sodium molecules. Holds on to sodium molecules, which in turn holds on to more water. Sodium attracts water. We have more water in the system, blood pressure increases. Now, as a result of holding on to sodium, it actually has to get rid of potassium. That's sort of the payoff that aldosterone has to do, which is why we release the antidiuretic hormone, which holds on to potassium as well. So we have an overall increase in fluid, we have an overall increase in sodium mostly, which gives you your thirst stimulation because of the high sodium content. You drink more and your blood volume increases as well. In the end, we have an increase in blood volume and an increase in vascular resistance, which in turn gives us a better blood pressure. Now, if it's the other way and it's too high, then this thing works by not releasing adrenaline. Short term is to instigate the parasympathetic nervous system. Long term is to limit the amount of renin, erythropoietinase, angiotensin 2, aldosterone that is released. Because this is naturally released throughout the body, but if your blood pressure is too high, it just doesn't release them for a while. Okay, therefore it allows your body to excrete, regulate itself back to a normal blood pressure state. And that is how we have the blood pressure controls within the body. So let's look at it all together. Now, I'm not going to go through this whole slide, but this is showing that what we learnt last time about the heart and what we learnt this time about peripheral resistance and blood pressure mediation control all work towards the same goal. And that goal is the presence of mean arterial pressure. Now, mean arterial pressure is another way of saying perfusion status. So somebody getting perfused means that the nutrients is getting to the organ bed. That's what perfusion means. And as we can see, the whole functions that we talked about, the unique structure of the heart, the special structure of the vessels, the homeostatic relationship with the rest of the body in measuring negative feedback systems, allowing for fluid retention, ionization retention, all of that plays a role linked in with the endocrine system and the central and peripheral nervous system, all to help regulate mean arterial blood pressure. Okay, so that is the cardiovascular system in a nutshell. Now, I hope you got something out of the second part. Obviously complemented the first part as well. I just didn't want to give you a really long lesson to listen to. But from here, you'll be able to use a lot of the knowledge that you've learned to definitely help you clinically and understand things like fluid therapy, things like pathological states, and overall vital signs that we use in order to measure what's going on in the body as well. So until next time, take care.